Teeth not only make our smile beautiful, but also their role in our digestion process is inevitable and of paramount importance. Otherwise, the human body would not be able to digest properly and result in severe indigestion. Teeth perform the first and pivotal role in digestion. They convert solid food into smaller pieces and, by further grinding the food, make it more swallowable and easier for the stomach to digest. With such importance in the life of a person to survive, because it is obvious we cannot live without food and enjoy the pleasures of life, one of life's most unpleasant occurrences is losing teeth. Losing teeth can have an impact on one's quality of life and self-esteem, in addition to their physical health. Stem cells are showing promise in regenerative medicine as an effective treatment for tooth regeneration, offering a potential new way to replace lost teeth. Stem cells are the actual healers of the body, and scientists are utilizing their healing ability to regenerate what is lost from a person's body. And regenerating the tooth is one of the services the stem cells are going to provide in the future of regenerative medicine. Stem cells are actually a type of cell that self-divide and can be differentiated into any type of cell in the body. This universal convertibility and their rapid growth into several billion cells of a specific type in the body are what make them unique and useful for regenerative therapy. Scientists are using stem cells to repair what the body has lost by utilizing their capacity to differentiate into any type of cell. For example, stem cells have been used to regrow teeth in mice, with similar techniques being researched for use in humans, and it is now possible, with supporting evidence and extensive research, for stem cells to regenerate lost teeth. For tooth regeneration, stem cells can differentiate into different types of cells that make up a tooth, including dental pulp, enamel, and dentin. Stem cells are capable of differentiating into each type of tissue cell involved in the formation of the teeth. This is a revolutionary development, as it allows for the possibility of regrowing teeth naturally, which would significantly improve oral health for people all over the world. The ability of stem cells to differentiate into these different types of tooth tissue is key to their potential application in the regeneration of teeth. The most prominent and therefore most potent stem cells for tooth regeneration are those obtained from the dental pulp of the teeth, the DPSCs. These dental pulp stem cells are located in the soft tissue inside the teeth, referred to as the dental pulp, and contains several multipotent progenitors that can differentiate into other cell types, like odontoblasts, which are responsible for forming the dentin and enamel of teeth. These stem cells can be easily cultured in the laboratory after being obtained from a patient's own dental pulp, making them the most reasonable stem cells involved in the regeneration of the lost teeth. Apart from DPSCs, there are other cells that also show potential in tooth regeneration, including embryonic stem cells, mesenchymal stem cells, and induced pluripotent stem cells. All these stem cells have shown great potential in tooth regeneration specifically. However, DPSCs have an advantage due to the fact that they can be obtained from a patient's own dental pulp, meaning that ethical and immunological issues can be avoided. Stem cell-based tooth regeneration is a technical process. In order to create a tooth, dental pulp, enamel, and dentin, stem cells must first be isolated from the patient's body or from a donor. Then, the stem cells must be stimulated to differentiate into these various types of tooth cells. The differentiated cells are then added to a matrix or scaffold that acts as a model for the growing tooth. The scaffold is implanted successfully into the patient's jawbone at the site where the previous tooth was lost. The stem cells there then continue the process of division and differentiation for the formation of various parts of the tooth. Over time, the newly formed tooth becomes fully functional and is integrated into the patient's mouth, and the patient successfully gets his own natural teeth. Though there could be further complications in the process, the potential is there, and the regeneration is certain. All that scientists need to do is improve and enhance the process and make it an applicable clinical therapy in the future. There are limitations in this process that need to be addressed to make it clinically applicable, such as the fact that it is a complex procedure, as there are multiple steps involved, and the coordination of several types of cells is also involved to enable the tooth to regrow and achieve full functionality. Likewise, the kinds of teeth that can be renewed might be constrained. Instead of entire dental arches, researchers are presently concentrating on the regeneration of individual teeth. The ability of stem cells to regenerate more complicated structures, like multiple teeth or the roots of teeth, is still unknown. As such, scientists must focus on further research and development to overcome the current limitations of this process to make it a viable clinical therapy. The potential of stem cells in regenerating teeth is positive, and the research is on the right roadmap. After achieving excellence, there will be a day when the whole world will have a smile full of teeth and no more toothless smiles.